Hello, everyone. Yes. So, first of all, I want to say it is a pleasure speaking to you today about a topic that is close and very important for me, and not only to me, but for a whole bunch of people. Some of them are actually sitting today in the audience, and I must say that this work has been the product of a year-long effort with scientists involved from 4 different continents. And before I start, I need to acknowledge that the work presented today has been conducted mostly in the ancestral, traditional, and unceded territory of the Musqueam people in British Columbia in Canada. And if anybody Googles jellyfish blooms, looks for nice pictures, of course, you would expect this very colorful, bright, shiny pictures of scyphozone and hydrozone blooms from the coasts, sometimes from the open ocean. But what you would never find is something from a lake or pond or river. And honestly, most people don't even know about freshwater jellyfish, and I'm sure not even all of you knew before this conference about it. But I'm actually very happy to see that 3 contributions in this symposium actually addressed this. It was the very nice talk by Sergio 2 days ago. It is now my talk, and there's a poster later on today also addressing freshwater jellyfish blooms. But before I go ahead, we need to kind of like set the stage what freshwater jellyfish are. And it's a very small group of hydrozoans that are medusa bodying, and they need to be separated from hydra and other non-bodying hydrozoans in limbic systems. As I said, it's a small group of organisms, 30 to 40 species about, and they occur globally in temperate to tropical habitats. And they mainly follow a typical metagenetic life cycle with a pelagic and a benthic phase. There's some shortcomings and there's some unknowns, but in general, this is what we believe is true for all of these. And when I talk about freshwater jellyfish, I concentrate on the 3 main genera, Astrohydra, Cuspidacusta, and Limnocnida. There's more, which I'm not going to address today. And some of these species actually became invasive, namely Cuspidacusta sovabi. And well, we have seen the symposium that we know quite a lot already about marine jellyfish, and we can't stop talking about all the limitations which doubtless are around. We know very, very little for the freshwater part. So, and I'm going to address some of the reasons why we know so little and why this is an actual problem. And so the reason is, why should we learn more about the freshwater jellyfish? Well, with climate change, we see that they are expanding their distributions. I will show you Meg in a minute. So we need to start caring soon before it's too late. And here are some more nice pictures of different species and life cycle stages from all across the world. But what is most impressive is actually the 2 bottom doesn't really work well. The 2 bottom pictures of blooms. The left one is coming from a lake in Canada, and then the right one is coming from Lake Tanganyika in Zambia. Different species, but same appearance. So one might ask, okay, when I say we have a hampered progress of knowledge gain in jellyfish, well, how does it really compare with marine jellyfish? What are the reasons for this hampered progress? And so if I say, well, it becomes an issue, where do we go next? What are the gaps we need to address? And very briefly, I want to give you an overview about the methods that we use to compile this massive data set. So we started with a literature search to see what is actually known already, classically with the Web of Science and Google Scholar, and used a couple of selection criteria, which I don't want to go into much detail now. But we very soon realized there's tons of articles that all our collaborators knew already, which weren't picked up by these search engines. So we decided to make a more exhausting by hand approach and came up with about 700 literature pieces in the end. And just to give you an impression how most of this year looked for me and several of my co-authors, so I went to many, many different libraries and looked up books that has been sitting in the shelves collecting dust for 100 years. Nobody looked them up. And even more exotic is the use of microfilms. I'm sure some of the older in the audience still know what microfilms are. I just learned to use them this year. All right, so we want to see how freshwater jellyfish research compares with marine jellies. And for that, I picked the best studied, doesn't really show up, it's too bad, the best studied freshwater jellyfish genus Crosspedacusta and compare the succession of research since 1880 to 2022 with that of ICAR-CMFRI, the probably best studied marine hydrozoan genus, and then the best studied freshwater hydrozoan genus Hydra. And well, the picture is quite different. So we see this near exponential number of publication compiling for Hydra because of obvious reasons, and we see a very different picture for ICAR-CMFRI, that basically until the late 90s, nothing so much really happened with the genus just after the discovery of the green fluorescent protein that we see this bump in the number of publications. But of course, it's very, very obvious that we know very little about Crosspedacusta, which is already the best known genus. All right, so what are the reasons for the hampered progress? And I just want to quickly walk you through the design of this plot in the bottom right corner. So we have the decade on the right, on the x-axis starting in the 1880s, and then the number of publications per year on the y-axis, and then color-coded in this example are the life cycle stages in question, frustules, medusae, and the polyps. And then on the right-hand side, the box plot basically compares the number of publications of the dominant life cycle stage, so the medusa, with the sum of all others. All right, but coming back to the reasons that basically hampered the progress. It is the misspelling and the change of species names. And this is not a very specific problem for freshwater jellyfish, but it occurred especially abundant in this. So it happened very often in the late 19th century that the polyp and the medusae stages were described separately, and people continued working with these different species in quotation marks, for years and decades until eventually they figured out, okay, the life cycle brings both species together and they needed to redescribe this. So there's a general impression of uncertain taxonomic entities. And of course, this is also not a problem for freshwater jellyfish only, but we see that the medusa stage is considerably better studied than everything else combined. So about 80% of the literature available on freshwater jellyfish is on the medusa stage because of obvious reasons. Well, the jellyfish is what the people usually find and has a potential harm for humans. It is also abundant in the literature that the research is very much biased towards one genus, Crosspedacusta. You can see it in the dark gray colors. And this makes very much sense, because where does this species or the genus usually occur? In temperate to subtropical regions, primarily in the northern hemisphere, and then in the countries of the global north, where actually people have enough resources to make research and publish this research. And it's also happened that this is the area on the globe, same on this, in the southern hemisphere, where you have the highest human population. So there's actually people around to make reports of sightings. And of course, stuff like geopolitical complications, wars, or objectively difficult areas make research on tropical species, especially in Africa, very difficult, that hampered the Limnocnida progress. And then another aspect that is not so much obvious for the marine jellyfish literature is the diversity in languages. While marine jellyfish research is most often in international waters, so it's obvious to use English as a communication language, this is not so apparent in regional scale and often national freshwater bodies, because the audience is most often a national dimension, so you publish in a national language. And if you look over the last 150 years, it's clear that in the first few decades, until about the 1920s, the colonial languages like English, French, German, and to a smaller degree Japanese were dominant. But we see also a very strong diversification in languages in then the first half of the 20th century, and especially after the dawn of the 21st century, two languages become dominant. It's English and Chinese. So for understanding freshwater jellyfish literature, it's important to not only read the English literature, but also to include other languages or literature published in other languages. And then there's, of course, a colonial economic crisis and global war signature in this general pattern that you see. Well, as I mentioned already, in the first 4 or 5 decades, we have a very strong colonial fingerprint basically on who conducted this kind of research. It was, of course, mostly the North Americans and the Europeans who had sufficient funding and who were the colonial nations to make this kind of research, to go on expensive long-distance travels, to collect all these data and eventually publish them in journals that are still accessible today. So publishing is not only the problem, it needs to be findable today. And so in the 1930s, we see a very strong diversification of people doing this kind of research on all continents, and then a very strong regionalization after the Second World War, which again hangs then together with the end of the golden 20s. Oh, sorry, now I mixed it up a little bit. Well, the golden 20s happened before the Second World War, apparently. Excuse me. So, but anyway, you see this in the 1930s, this very high output of papers, which then plummeted in the 1940s related to World War II activities, and then this uprising again in the 1950s, which is of course related to an increase in funding for fundamental and applied research, not for freshwater jellyfish only, but in a global perspective because of a nuclear potential threat. And one aspect that we shouldn't underestimate is the complex between science and military. In many marine sciences, we as scientists piggyback on marine military vessels like the Coast Guard or Navy, which facilitates our research as an expensive platform and waives a lot of our expenses. But nothing like this exists in smaller freshwater, often within national boundary water bodies. So this kind of research faces very strong financial challenges. And then lastly, there's a lack of theme-led journals where actually freshwater jellyfish research is published. And I summarized all these literature and found that only 15% are published in 5 primary outlets. Well, there's Science and Nature among those. Obviously, very few people can publish freshwater jellyfish research these days in these journals, but this is abundant in the early decades. And again, this strongly contrasts with marine jellyfish research, where we have well-known journals. And all of this contributes then to the impression, well, there's little progress, there's little need, so why should we care? And the reason why we should care is actually these 2 maps. On the left-hand side, you see the present day. On the case, and the right-hand side, you see the 2050 case, and it's a species distribution model for the most abundant and west-studied species, Crossbetta-Costesova. And what I can basically say as one just sentence, that over the course of climate change, this species and potentially many other freshwater species will expand the distribution. So whatever they do, we need to start caring and understanding before we actually can start managing them. And because I'm running short in time, and this is a very extensive list of things that we actually don't know, and I'm sure every one of you will find some of these aspects reflected in their own work, well, this is the open field. Feel free to join in. It's basically each of these points has 1 or 2 papers historically, and we know literally very little about where they are, what they do, how do they distribute, how can they be managed, what effects they have on fish, tourism, how they interact with other food web components, and so on and so on and so on. All of this is detailed in the associated paper, and I can't go through this all right now. But what I want to say at the end of my talk is that all this literature has been summarized in this Pangea data set that is also freely available already, so you don't need to start from scratch again. All of this information has been summarized, is accessible now. And as a very quick take-home slide, I know it's a lot of things to take in, 150 years compressed in 12 minutes. It's hard, but our present perception is governed by one genus only. There's more. All of these different genera and species do different things, which we have very little idea about. If the researcher is only reading English literature, It's apparently missing about half of the literature available, so it's important to group up in international teams. And we need especially to put forward the knowledge gain in Africa, Oceania, and South America in collaborative efforts, because these are the areas where we know the least. Thank you for your attention.